So here we have you want to use the pointer?
Yep. So we'll switch from here.
Okay. And this one?
Yes. You told me this has to be in the front.
It's better outside, yes. Yes, and then this one, exactly. Something.
I think it will work. Now it's all too strong. Can you hear me? Can people hear me? No, okay, it works.
So let me tell you at 5 minutes or some time for questions.
Quarter to 10.
Quarter to 10. So I will tell you in 5 minutes.
Oh, just tell me quarter to 45 tell me. Yes.
Okay, so we will continue with this last section of today because we have had this time. So now our next speaker, most of the people already know because she is the coordinator of this IPL, or the ALLODD, so Cournia. So she already did the thesis in Heidelberg, then she did the postdoc stay with Jorgensen in Yale.
Thank you very much. It's wonderful to be here, of course. Thanks to the organizers, Xavier and Carlos, for hosting this event and for allowing me to give this presentation, which is kind of a summary. When I mean, all the talks were going on, and I was realizing, oh, this is in my slides, this is in my slides. So it's a great opportunity to summarize what we have heard today, possibly some tomorrow. So I will talk about Allostery and Drug Discovery and how we can merge the two worlds. So at the current time, we have the era of precision medicine, which means that all the current drugs, or most of them, are discovered taking in mind stratification of patients and different, for example, mutations that they have if they have cancer, so that we can separate them in different groups, so patient stratification. As, for example, the very famous example of lung cancer, where it was found that 4% of cancer patients with non-small cell lung cancer have a rearrangement in anaplastic lymphoma kinase that leads to carcinogenesis. And then drug discovery, drug design for the specific subset of patients that have this rearrangement was performed, resulting to crizotinib for people who have this particular rearrangements. And the cornerstone of this approach, rational drug discovery for precision medicine, is that we can utilize three-dimensional structures of proteins in order to target a pharmaceutical target, commonly a protein, with small molecules. However, most drugs are designed to bind directly to the active site, which very often leads to off-target effects when proteins are similar, have similar binding pockets. And also, we have one or at most two active sites in the protein, which leads to ideas falling short, coming through the patent cliff, where basically pharma is running out of ideas. And as the patents expire, we need new technologies to start modulating old drug targets, old proteins, but also drug targets that are currently considered undruggable. So allostery could be a new strategy for drug discovery. So small molecule drugs that can be designed to bind into secondary binding sites, as Jean-Pierre said, it's a Greek word, so we're very proud, so it means different sites. So basically, the allosteric modulators bind at different sites and modulate the protein from a distance. So the evolution of allostery, we heard that in 1961, it was the first time that the term allosteric was introduced. And going on to the evolution of this history, the first allosteric drug was approved by the FDA in 2004. And perhaps we're discussing that perhaps allostery is an intrinsic property of all proteins or proteins with dimeric or multimeric partners. And it can be proposed that allostery can be utilized as a new drug design strategy. So essentially, the TEMs and the relaxed states or inactive and active states are properties of conformational ensemble. So they are all structures that lie in a conformational ensemble. And a perturbation at an allosteric site would lead a shift to the occupancy of states by the population. For example, if you have an agonist that will stabilize the free energy landscape of the active state, while if you have an antagonist, it may stabilize the TEM state, the inactive state. So we can say now that allostery is a property of the conformational ensemble. And in this framework, we can consider that the allosteric pathways, so allosteric modulators, regulate the activity through pre-existing allosteric pathways. So all these connections of the amino acids that can regulate the allosteric site from a distance, they could be pre-existing. So when an allosteric activator, for example, binds in an allosteric site, it could facilitate more efficient substrate binding through these pre-existing pathways. Or on the other hand, when you are binding an allosteric antagonist, then you could have the effect of not binding the substrate. So just to give some basic ideas of or concepts of the effects of allosteric modulator within a receptor, that they are important for you because I will talk about them later, is first to increase or lower the affinity of the orthosteric ligand. So it means how much the orthosteric ligand is binding, how much it's occupying 50% of the targets at equilibrium. Then we have the concept of efficacy. It's not only the affinity, how much it will bind, but also the efficacy, which means the ratio of desired response to required concentration of the ligand. So how much the reaction, let's say, is happening. So acting as an agonist will activate a receptor to produce a biological response, and acting as an antagonist will block the binding of other allosteric modulators. So we've categorized the types of allosteric modulators, if we can categorize them somehow. Tried really hard. I hope this doesn't omit any classes. To positive allosteric modulators or PAMs, we've heard also from Brian. Negative allosteric modulators or NAMs. Silent allosteric modulators, and I will go in each one of the categories to explain what they do. Allosteric full or partial agonists. And then we have the Ago-allosteric modulators, which can also be PAM agonists, PAM antagonists, and NAM agonists. I know it sounds very confusing. I was also very confused when I was reading this, but I hope I can give you some feedback on that. So here again, we will play with the affinity, the efficacy, affinity of the ligand, the orthosteric ligand, the efficacy of the orthosteric ligand, agonism and antagonism. So this first class, the positive allosteric modulators, increase the affinity of the ligand at the orthosteric site, so how strongly it binds, but also the efficacy of the endogenous agonist, so how well it performs the biological function. The negative allosteric modulators, they would have the opposite effect, so they will lower the affinity, essentially not allowing so efficient binding of the orthosteric ligand. but also their efficacy. They will also lower the efficacy of the endogenous agonist. Then we also have the silent allosteric modulators. And also may I say that don't have to take notes. This will be recorded and will placed in the ALLODD YouTube channel. So the silent allosteric modulators, they don't affect agonist activity of the allosteric ligand, but they actually can act as an antagonist to other allosteric modulators. So essentially, they will antagonize with other allosteric modulators, but will have no effect on the efficacy or affinity to the allosteric side ligand. Sorry. And then we have perhaps the most complex category of allosteric modulators, which are the Ago-allosteric modulators. So these types of allosteric modulators can act at the same time as modulators for endogenous ligands, as well as allosteric agonists or antagonists, even in the absence of allosteric ligands. I know it sounds very complicated. I will try to explain it as much as I can. So here we have a positive allosteric modulator, which is also an agonist. It means that it will enhance the affinity and the efficacy of the allosteric ligand. Okay, that's pretty clear. But then when you have a positive allosteric modulator, but an antagonist, what does it mean? It means that it will enhance the affinity of the allosteric ligand, but it will reduce its efficacy. So you can have crazy things happening. So here, this Ago-allosteric modulator, the PAM antagonists, increase the affinity of the allosteric ligand, but reduce its efficacy to perform a certain reaction. And then we also have the negative allosteric modulator, which are agonists. Here, what's happening is that the Ago-allosteric modulator reduces the affinity of the allosteric ligand, but it increases, so this is wrong, it actually increases the biologic response, the efficacy. So this arrow should actually be up. So we have many different combinations of things that can happen with allosteric modulators, and that's one of the really exciting facts of this type of modulation. And we have three different types of cellular receptor families that can be allosterically modulated, and we know that. So for example, the G protein coupled receptors, we've heard about them, channel-linked receptors, such as the nicotinic acetylcholine receptor, Jean-Pierre very beautifully illustrated, the receptor tyrosine kinases, and of course, intracellular receptors. So I'm going to try and give some examples from those. So one famous example, and this was relating to the question of Marco Cecchini to Francesco Gervasio's presentation about, oh no, that's the PIF pocket, that's my next slide. This is not related to that, it's coming up. This has to do with the SHP2 protein and inhibitors of those. So this is an oncoprotein, and at the same time is an immune modulator at the PD-L1, PD-1 pathway. And this study by Novartis, it actually showed that you can have simultaneous allosteric inhibition at two sites. And it was possible to develop compounds, allosteric modulators with very good pharmacokinetic profile and selectivity, which has been a very challenging case for phosphatases. And now it comes the example of the PIF pocket. And of course, the presentation crashed. It has to be related with Francesco, I'm sure. Okay, let me go back to my slide. Okay, great. So this is a very exciting case of allosteric modulators. because with the same, we have here the allosteric ATP pocket. Okay, this is ATP. And then we have an allosteric pocket that was discovered, the fifth pocket that was described by Francesco. And it looked like that modulators binding at the same allosteric pocket different ones could actually, some could actually activate the protein and some could inhibit the protein at the same site. So this was what Marco was asking. So allostery can both activate and inhibit PDK1. Here we have the allosteric activator A2A, that is right here, that enhances the efficacy of the protein. So it's an agonist. But at the same time, if you bind a molecule which is called IF8 at the same pocket, and you can see both of them are superimposed, so very close, it triggers a conformational change in the ATP pocket, acting as an allosteric inhibitor. So PDK1 is a great example of allostery because it can show us that at the same time we can have both types of modulation, but also because it has been really difficult to show activation of kinases in any other way. So there's another quite famous example of allostery with new properties. And this is a study published in Nature in 2017, where it was shown that treatment of BCR-ABL mutant with an allosteric inhibitor could overcome drug resistance. So it was known that when you bind nilotinib in the active site, actually a lot of mutations start to evolve after continuous treatment of the kinase with the allosteric ligand, nilotinib. But when you co-administer an allosteric inhibitor at a mid-story site, ABL01, at the same time with nilotinib, then the drug resistance stops completely, and you don't have a resistance to the actual drug, which is a real breakthrough. And we think that this could be applied efficiently to other cancer drugs with co-administration. So I showed some examples of proteins that are soluble, and now it's time to move on to the membrane proteins as allosteric bodies, because they're actually one of the biggest drug target classes. And we've heard about GPCRs, of course, one of the largest family, a wonderful talk by Brian. So I won't go through that. Although there's activation, we've heard of numerous signaling pathways, equally potentiating or inhibiting the signaling pathways with different modulators, selectively modulating specific signaling pathways, and having little or no effect to others. We've heard that there are so many allosteric pockets that could be exploited on GPCRs, many of them unknown, and many ligands that have actually no characterization as allosteric yet, but they could be allosteric. And also heterodimerization of GPCRs driven by allostery is also a significant potential drug target. The ligand-gated ion channels, of course, I've heard from Jean-Pierre, the central role in the function of the central nervous system. And we've heard about the blooming and twisting, and actually how with specific modulators, here agonists, you could stabilize the resting state. And here in complex with ivermectin, again, you can stabilize the resting state. We've heard about receptor tyrosine kinases that are an integral part of signaling because they function as receptors to growth factors, cytokines, and related hormones. And Francesco gave you extremely good examples, so I won't spend more time than saying that normally the epidermal growth factor, which is a hormone, binds onto EGFR and dimerizes, so facilitates the dimerization of the protein, which triggers a response. And an antibody can allosterically bind to a different than the active site, allosteric site, and then it could stop the dimerization by freezing the protein in certain conformation and stopping the biological response. And a topic, so some classes of proteins that are very close to the interest of my lab are peripheral membrane proteins and lipid anchored proteins. And actually, the first drug to ever target KRAS, which has been the holy grail, and nobody could actually develop a drug for that. The first drug that was marketed in May of 2021 was a KRAS-4B G12C mutant allosteric inhibitor. Here is the GTP, so here is the active site. And the molecule that was discovered, an evolution of AMG 510, it actually an evolution of ARS 1620. It actually binds on an allosteric pocket, but if you could expand the actual molecule, it would open up a cryptic pocket next to the allosteric pocket to even further enhance the activity of this ligand by a new molecule opening up this cryptic pocket. So this was the first ever marketed drug for KRAS-4B. So the FDA approved in May 2021. Another peripheral membrane protein that we work on in my lab is phosphoinositide 3 kinase alpha, or PI3K alpha. And we have studied this with experiments on SPR, so binding essentially the liposomes on an SPR chip, and then throwing on top on the solution PI3K, and then measuring protein membrane binding, where we find that actually a mutant that's very far from the active site is binding to the membrane much more efficiently than the wild type. And then there's other classes of mutations like the E545K, which we studied together with Francesco, and he talks about that, so I won't dwell on that. But with computer simulations together with biophysical experiments, we can facilitate a molecular level picture of the associated mechanisms of the mutation, which are allosteric, because none of these mutations to PI3K are in the active site. They're more than 20 angstroms apart from the active site. They're very far from the active site. And here with Francesco, we show the different conformational energy landscape that occurs when you have the mutant E545. So together with the computations that I showed for the 1047R mutant and the computational experiments we did for E545 covering the free energy landscape of the mutant, we set out to do a drug screening campaign with a binding site prediction, starting from crystal structure and extensive molecular dynamics that uncovered the full free energy landscape, as Francesco very nicely mentioned. Then finding binding sites that were there on the protein, but actually not all the binding sites that you can find on a protein are allosteric. Maybe they are there, but don't do anything. So through initial, let's say, basic techniques, initially we confirmed that these pockets were allosteric because they would communicate their motions to the active site through cross-correlation of the residue. And after we confirmed computationally that these pockets are allosteric, we did a screening, a virtual screening, a normal virtual screening exercise with GLIDE, post-processing compounds, binding them, and doing the in vitro experiments. And as I mentioned, here's the active site, here's the mutation site, and this is one example of a pocket with actually a crystallized ligand, which, however, is not allosteric. So not everything that you see bound to a compound means that it has an allosteric effect. The allosteric effect needs to communicate, to propagate a signal to the active site and modulating the biological response of the activity of the ligand. Otherwise, it just sits there and it may not do anything. So through in vitro experiments, we tested different concentrations of ATP to validate the activity of kinases, and we found that our modulators, the molecules that we designed through the virtual screening that I described, actually did not change their activity when we are changing or even flooding the system with ATP. So essentially, they remain inhibitors, contrary to non-competitive inhibitors of active sites. So these molecules could be considered allosteric. And then we did a series of experiments, but most importantly, in vivo experiments in mice, where we treated with these compounds PI3K wild-type tumors. So here the cancer is not caused by PI3K mutation, and tumors that have the PI3K mutation. So when we treat with our molecule, PI3K41, tumors that have nothing to do with PI3K mutation, there is no effect in the tumor. So you see here the tumor volume. Even if you treat the tumor with water or with a molecule, it really doesn't make a difference. If you move to tumors that are dependent on this mutation, and because we have designed the drug candidates with in mind the allosteric modulation of E545K tumors, you see that there's a dramatic effect on the tumor volume with respect to the vehicle, which is water. So you see here that it was possible to design modulators that selectively inhibits the E545 mutant with respect to the wild type. But even more importantly, we wanted to understand how these molecules are acting. And here we try to understand the biophysical picture of what is happening to the protein. And that relates to the very nice presentation that Miguel showed us earlier, where essentially you can measure with SPR the binding of the protein on the membrane. And in his case, he finds that when he binds the molecule on the protein, when you try to bind it to the membrane, the protein-membrane interactions is completely disrupted. And in this case, we actually find that again, there is a very big effect with our allosteric inhibitors on protein membrane binding. So when we monitor membrane binding with a protein in the presence of a competitive inhibitor, which is wortmannin, we see no effect. So this is a difference in membrane protein binding for wild type and mutant. So no effect for a competitive inhibitor. But when we actually bind any of our molecules that we design as allosteric, we see huge effects on the protein membrane binding. And in our case, it's not a protein membrane binding inhibition, but it's rather an agonism. So the protein binds more stably to the membrane, possibly in an inactive conformation. So that tells us that these allosteric modulators, they function with a way that it modulates the protein membrane interactions. So here we have discovered the isoform and mutant specific PI3K alpha inhibitors that modulate protein membrane interactions. And because we're very interested in these allosteric mechanisms in membrane proteins, and we wanted to take it a step further into opportunities for drug discovery at the protein membrane interface. Because these proteins that are important drug targets and they are associating with the membranes, we believe that we can modulate their interactions by altering the protein membrane binding. And this is an example that Miguel suggested, that having an inhibitor could actually modulate the protein membrane interface, in this case inhibit, in our case to enhance the protein membrane interaction, but with an inactive state. So our workflow for targeting protein membrane interfaces as a drug discovery or drug design strategy is to identify membrane binding domain with an algorithm, and I will speak a little bit about that, generating a protein conformational ensemble, identifying binding pockets at the sites of where the protein attaches to the membrane, assessing whether this binding site is implicated in allosteric networks, and then assessing next if this site is druggable on this protein membrane interface, and then performing a usual drug discovery campaign in these cavities, and of course in vitro and in vivo assays. Okay, so I have still 5 minutes. So basically what we want to achieve is to test whether, how or where the protein will bind on the membrane by identifying the membrane binding sites, then by identifying new cavities at this interface that could be exploited for drug discovery, and then most importantly, to understand the allosteric, these pre-existing allosteric networks that would connect our cavities to the active site, because if they are connected, it means we could have allosteric modulations. So we created a workflow for targeting these protein membrane interfaces. So we built an ensemble machine learning pipeline using structural data from the protein data bank to predict these protein membrane interfaces, which is published here. But then we also built a platform, which is called DREAM, that you can use to predict these pockets, binding sites and protein conformational ensembles. And as mentioned, we've implemented this as a web server. The paper is under review. And we have also onboarded it in the European Open Science Cloud. So it's an open source and open access code that you can use. And to finish off, let me say that allostery may be a general feature of proteins. This is to be discovered. We have a constant growth of the discovered allosteric inhibitors. So you see here by very, very few, really an exponential growth in the last years. We have technologies that can discover or unveil these cryptic sites. For example, a combination of experimental techniques such as fragment-based screening with computational techniques such as MDMix of the Barril lab, where you can unveil these cryptic sites, which could act either through conformational selection or through induced fit, or through both conformational selection and induced fit. So the allosteric modulators can bring a real evolution, we think. They target sites that are less evolutionary conserved, so greater protein subtype selectivity can be achieved. You can selectively target proteins, non-competitive endogenous ligands. We have an opportunity to exploit all targets with new chemical space and to do a combination therapy with allosteric sites that can limit the resistance profile, as I mentioned. Target proteins that have been considered undruggable, such as KRAS. And protein interactions, as Ben told us, are very difficult to inhibit with allosteric drugs. So allosteric modulators present also opportunities for protein-protein interaction modulations. Of course, as every discovery doesn't come without a challenge. Challenges of allosteric modulators include that they're usually shallower and narrower than allosteric sites. Minor differences in allosteric modulators, as the example that I showed you, can lead to opposite effects of the protein function. So for the PIF pocket I showed you, we had an agonist and an antagonist in the same pocket, so this may be challenging. Allosteric sites are often unknown or difficult to predict. And the less evolutionary conserve among such subspecies could also be a challenge. This is a review that we wrote with Alexis Tadjoulas on the rational design of allosteric modulators, just in case you're interested for more reading. So we have covered many different cases than those that I had the time to present today. So just to close and finish and take some questions, we really believe that allostery can emerge as a very promising avenue for creating novel modulators of membrane protein function. And I really think that the new classes of drug therapies will come from allosteric drugs. So thank you very much. This is a very nice consortium we have been working with, Allostery and Drug Discovery, or ALLODD, 18 partners from many different countries. And of course, I would like to thank my group and also some collaborators that did the experiments. And Bruno, who is here, is my SR, who will work on discovering the allosteric pathways. Of course, my collaborators, Francesco, is here. Okay, I fixed it here.
Thank you very much, sir. So we have some 5 minutes.
Thank you very much for the very nice talk. One comment and one question. So the comment is among the challenges, I think we can design positive and negative allosteric modulators, so eventually look fragments and antagonists, but we need to be better precise regarding range interpretations. And in the case of the glycine receptor, which is the system of studies, so with the channels, give a full agonist and very partial agonist. The delta G's that we have to capture is to become more.
Wow, that's at the limit of
we
reality.
be extremely precise, and that's of course the challenge of saying a system is so flexible and now the question is, I was very curious about drug resistance when you
Yes.
use example of kidney
Mhm.
in combination with the modulator. Can you explain me why we avoid drug resistance when you have complication? I mean, is there any evidence for this?
No, well, there was this paper that was published in Nature. I don't know if anybody has more information. But as far as I understood, they didn't really uncover the mechanism of how this happens. But what they observed is that when you have the dual inhibition, you limit the resistance profile. But maybe you have more information.
With antivirals and with cocktails, they usually go to different types. When you have a cocktail of drugs that goes to a safe type, you get the same effect.
This is an inhibition. Yes, inhibitor, inhibition. Inhibition of the. The mutations activate the receptor, so this chimeric BCR-ABL protein, and the molecules act as inhibitors of the mutation, not of the mutation, well, yes, of the chimeric protein, which is the BCR-ABL in leukemia. Yes.
Species and sort of complementation pathways that impact the circumstances. What are the last features in the one of the units in memory and I've got some experimental data that's not been published, but obviously sometimes that lipids a lot of time size that can be acid modulated. And we know that some lipids sometimes get modulated in
Exactly.
silencing those. So with the method that we're using computer chemistry, we tend to homogeneous lipid bilayer. Is this something we're looking at?
Yes, absolutely. So thanks for bringing it up, because when I finished the presentation, I realized that I didn't include this type of inhibition, but of course, Brian showed it. So yes, we know that lipids and cholesterol, they bind to allosteric sites and modulate protein function. And these sites, they could be used as allosteric sites for protein modulation. So it is very crucial, and my lab is working very actively on that to model realistic bilayers as much as possible by including the different species of lipids, sometimes including 2, 3, or 4 and lipid species. And of course, that sounds very nice, but in reality, it's very difficult to equilibrate mixtures of many different species, which is up to the microsecond or multiple microsecond time scale. That has been very challenging. And on that front, what we are doing is that we are equilibrating in coarse-grained representation and then reverting back to back mapping the equilibrated bilayer in order to proceed with the calculation. Yes, so that's a very, very important point. And to capture these effects, I think you really need to have a realistic bilayer. And there have been reports in the literature where having the incorrect bilayer completely misses the effects of allostery from within the membrane. So thank you.
Yes, thank you very much for your talk. I was wondering, let's say you have identified an allosteric site and you want to launch a protein for that. For example, is the way to go beforehand if you will have if the ligand will have an agonist effect or
Very good question. I don't have an answer. I don't know if Brian has an answer. He doesn't. I don't think anybody has an answer, but maybe with Ben's technology and the technology that we hope to develop in the ALLODD consortium, we can predict these effects more efficiently. This will be the goal of this consortium. Somehow to bring in different experimental and computational techniques, sorry about that, of course about practice, to understand the biological response of allosteric ligands and understand exactly what you said, to predict if a molecule will be agonist or antagonist or any other of the different types.
Okay, so thank you very much, Zoe.
Thank you.